The max cytokine secretion assay technology allows detection of secreted cytokines on the single-cell level and sensitive isolation of viable cytokine-secreting cells. In order to label IL-17-secreting cells, a single-cell suspension of mouse leukocytes is prepared and stimulated at 37 degrees Celsius with PMA ionomycin to induce cytokine secretion. To stop secretion, cells are then placed on ice where they are exposed to the IL-17 catch reagent. Once secretion is reinitiated by increasing the temperature to 37 degrees Celsius, IL-17 is trapped by the catch reagent, a bispecific antibody that binds to CD45 on the cell surface of leukocytes and to IL-17 as it is secreted and caught near the cell surface. Secretion is then stopped again by placing cells on ice. To detect the trapped IL-17, cells are incubated with a second IL-17 specific antibody conjugated to biotin and an anti-biotin PE antibody. Cells can now be directly analyzed by flow cytometry or prepared for isolation and enrichment by subsequent labeling with anti-PE conjugated microbeads.
Hi, my name is Elly Rankin, one of the scientists at Miltenyi Biotec. Today I'm going to show you a procedure how to isolate and detect Th17 leukocytes, which upon stimulation secrete IL-17. IL-17 plays an important role in the adaptive immunity and inflammatory response.